Assalamualaikum and a very good day. I am Muhammad Ahmad Putri bin Ma'rifat from AS2556A and I am delighted to present my thesis project entitled Antibacterial and Antioxidant Activity of the Kratom Extracts Mitragyna Speciosa. This video referred to the table of contents which consists of introductions, literature review, methodology, result discussions and also conclusions. Moving to the first phase, which is introductions, Kratom, which is also known as Mitragyna speciosa, is a tropical deciduous tree native to Malaysia and Thailand, with leaves known also as puric in indigenous cultures. Its leaf contain over 25 alkaloids, particularly mitragynin, known for medicinal benefits like antidepressant properties and also alleviating alcoholism symptoms. Research suggests that kratom leaf extracts have potential as cost-effective alternative antibacterial compounds against antibiotic-resistant bacteria. This study aims to evaluate secondary components in kratom leaf extracts and also determine the optimal dosage to eliminate gram-negative and also gram-positive bacteria and explore its antioxidant activity. As for problem statement, traditionally, Thai and Malaysian laborers use kratom as a stimulant to combat fatigue and muscle strains. Identifying kratom's active compounds is crucial for the pharmaceutical purposes. An experiment using kratom leaf extract from UiTM Pahang branch Jengka campus can assess its antibacterial activity against gram-positive and gram-negative bacteria via agar well diffusions. Furthermore, the antioxidant properties of the kratom extracts can be studied. Antioxidants protect cells from the oxidative damage, potentially reducing the risk of disease like cancer and also heart disease. For significance of the studies, kratom leaf has traditional uses as one poultice, fever treatment, muscle pain relief, appetite suppressant, and also diarrhea. Utilizing kratom's potential for bacterial treatment and resistance prevention is crucial. Kratom products have expanded to pills, capsules, powder, and also drinks, creating opportunities for the medical development. Understanding its antioxidant and active compound will aid further research and analysis, serving as valuable reference for the future studies. The thesis project consists of 3 objectives, which are firstly, to identify the part of kratom with a high active compound. Secondly, to measure the antibacterial activity of kratom leaf and to analyze the antioxidant property of kratom extract. For literature review, kratom, which is scientifically known as Mitragyna speciosa, is a tropical evergreen tree native to Southeast Asia Pacific. It belongs to the Rubiaceae family and shares a close relationship with the coffee plant. There are 10 species in the Mitragyna genus, with 6 found in the South and Southeast Asia and 4 in Africa. Kratom leaves are elliptical with dark glossy green color and 12 to 7 pairs of veins. The flowers are arranged in 3 head grouping and the fruits contain several flat seeds. Kratom is traditionally consumed by preparing decoctions or teas from the leaves, and it can also be dried and processed into powders, capsules, and extracts. The main active compound in kratoms are mitragynins and 7-hydroxymitragynins, which act as an opioid receptors in the brain. Kratom's effect can be stimulant-like at lower doses and sedative at higher doses. Different strains of kratom can be distinguished based on leaf color and origin. Kratom has been used traditionally in Southeast Asia for various purposes. It has pharmacological effects including analgesia and suppression of opioid withdrawal symptoms. However, clinical research on kratom is limited. Kratom extracts have shown antibacterial properties against certain bacteria, and they also possess antioxidant characteristics, making them potentially beneficial in phytotherapy. The antioxidant activity is assessed through methods like DPPH test. Overall, kratom's potential pharmacological properties and its active compound have sparked interest in its usage, but more research is needed to fully understand its benefits and risks. Moving to the methodology, it consists of 6 steps and methods, which are direct observations and literature review, preparations of the samples, maceration method, phytochemical screening, agar well diffusion method, and lastly, the DPPH analysis antioxidant activity test procedure. For direct observations and literature review, 3 samples of kratom plants were harvested and classified based on their stages. Sample 1 is from 9-month-old saplings, while sample 2 is from the middle part with white and healthy leaves, and sample 3 from the base with the older leaves. The collected samples were from plants aged between 4 to 5 years. For preparation of samples, freshly harvested kratom leaves were washed, drained, and weighed for their wet weight. Afterwards, they were dried until fully crumbly and reweighed to determine their dry weight. The dry leaves were then ground into a fine powder and subjected to the maceration process for extraction. As for maceration method, kratom leaves weighing about 35 grams were mixed with ethanol solvent in a sealed beaker and kept away from direct sunlight for 5 days with periodic stirring. The filtrate was separated from the pulp. The pulp was then extracted again with the remaining ethanol and left for 2 days. Filtrate 1 and filtrate 2 were then combined and evaporated using a rotary evaporator at 68 degrees Celsius, close to the boiling point of ethanol, which is 78 degrees Celsius, in order to obtain a thick ethanol extract of kratom leaves. For phytochemical screening, the kratom leaf ethanol extract underwent various phytochemical screening procedures to identify specific compounds. The screening methods included wetness reagent test for alkaloids, foam test for saponins, alkaline reagent test for flavonoids, Bremer's test for tannins, and also Sarkowski test for terpenoids. These tests help determine the presence and evaluate the composition of the extract. For agar well diffusion method, the agar diffusion technique was used to test the antibacterial activity of kratom leaf extract on the nutrient agar medium. Paper discs soaked in the extract were placed on the agar surface, and the sample was then incubated for 24 hours at 37 degrees Celsius. The inhibitory growth zones were measured to determine the antimicrobial activity using E. coli. Distilled water and antibiotic discs were used as a negative and positive control respectively. As for DPPH analysis antioxidant activity test procedure, the DPPH radical scavenging assay was used to evaluate the effectiveness of kratom extract in neutralizing the free radicals. The extract was mixed with a methanol solution of DPPH, and then the color change was observed. A spectrophotometer measured the absorbance at 570 nanometer after 30 minutes of reaction time at the room temperature. For results and discussions, let us go through the phytochemical screening first. For wetness reagent, the wetness test yielded positive results for all 3 samples of kratom, indicating that the presence of the alkaloids in each sample. The reddish-brown precipitate formed during the reactions is a characteristic indication of alkaloid presence formed through the covalent combinations of potassium ions with nitrogens in the alkaloids. As for the foam test yielded positive results for all 3 samples of kratom, indicating that the presence of saponins in each sample. The formations of the foam after boiling and shaking the samples for around 5 minutes is a characteristic indication of saponins present in the kratom samples. As for alkaline reagent test, the alkaline reagent test showed no presence of flavonoids in the kratom samples. Sodium hydroxide was added to the extracts and no strong yellow color formation occurred, indicating that the absence of flavonoids. The color did not change even after adding diluted acids. As for the Bremer test showed the presence of tannins in all the 3 kratom samples. When 0.1% of ferric chloride was added to the extract, a brownish green or blue-black precipitate formed. By this, it confirmed the presence of tannins. And lastly, as for the Sarkozy test showed no presence of steroids and terpenoids in all 3 kratom samples. The absence of a red color in the bottom layer and a yellow color layer confirmed that the negative results for these compounds. Next, let us move to the agar-well diffusion method. In this study, 3 samples of the kratom leaf extract were tested for antibacterial activity against E. coli. Higher concentration showed stronger inhibitory effects, reaching a plateau at about 18 to 100%. Kratom's alkaloid may disrupt bacterial cells membranes, leading to cell death. For this, E. coli was used as an alternative due to the unavailability of the Aeromonas hydrophila. As for the DPPH analysis antioxidant activity test, the study evaluated the antioxidant activity of 3 kratom samples at different concentrations. The highest concentration showed the highest absorbance and IC50 value, indicating that the potent antioxidant activity. However, at very high concentrations, the antioxidant effect may become less efficient. The limited samples availability hindered precise classification of the samples. Variability in the results might be due to the errors in sample preparations, outdated spectrophotometer, and lack of standardizations. However, despite these limitations, the study confirmed the antioxidant potential of the kratom. In conclusion, this project aimed to identify the highly active compound in kratom, measure its antibacterial activity, and analyze its antioxidant properties. The study revealed that kratom contains active compounds such as alkaloids with positive results in antibacterial screening. The antibacterial activity was found to be concentration dependent, with higher concentrations leading to stronger inhibitory effects. Additionally, kratom extract also demonstrated antioxidant effects, but limited sample availability affected the conclusive analysis. Despite this promising finding, further research is necessary to understand the mechanism, safety, and therapeutic potential of kratom and its compounds for clinical use. Okay, that's all from me, and thank you for lending your ears.